with. So this is what we call the dogma of biology. And we all know what DNA is. And that's actually consists of this code of letters. And they match each other. As you can see, C's go with G's, A's go with T's. And this is what gets replicated throughout generations. However, something really beautiful that happens, it's something that this machinery comes, and the one that replicates DNA, and it doesn't replicate it exactly alike. It makes mistakes. And those mistakes are the reason why you're sitting there, right there. It's the biggest driver of evolution. If it wasn't for that, we would all still be single-cell organisms. So it's a very important thing that makes that mistake. Now, from DNA, there's another machinery, a little machine called RNA polymerase that comes and creates mRNA, which you might have heard from the COVID vaccine. And this is complementary to where it's reading. So it can read in either one of these strands, that machine. And the complementary to where it's reading, it creates this RNA. That RNA will leave the nucleus of the cell, go and another machine called the ribosome will read this code, and every three of these letters it'll tell it basically go and get this amino acid. For example, here it's methionine. Then the next one it'll attach will be proline. So each one of these stands for a code. Just keep that in mind. And then these amino acids together form a protein. And depending on the properties of each one of these amino acids, it will make a three-dimensional fold and will give it the characteristics. And that's the difference in proteins, the diversity that we have. This kind of representation of what it might look like, the machine itself. So you see the DNA, it's unwinding it here, and the messenger RNA is coming out. Now, genes, they're not all turned on at the same time. We have to have some kind of system of control. And this is what we call epigenetics. And there are many different ways that we know that gene expression is controlled, and there's some ways that we still don't know. We're still trying to figure it out. Now, one of the main ways that's going to be important for this presentation that you understand is something called DNA methylation, which basically on those C's, this DNA methyltransferase, a different kind of protein, will just add a little molecule right here, and that's methylated, and that's depending on the region, can turn on a gene or turn off a gene. As you can see, DNA also wraps around these proteins called histones, and they have these little tails that can also have changes, some methylation or acetylation, and that causes them to either unwind or wind and give access to DNA to be transcribed. Another way is something called long non-coding RNAs. When we first mapped out the genome, they thought that most of it was junk DNA, and we come to find out that there's no such thing as junk. It's there for a reason, and a lot of those that we thought were junk are actually part of the regulatory system. And then it's really important that you understand a couple of things. There's a protein called CTCF, which makes these chromatin loops. So as you see, the DNA was wrapped around the nucleosome, and that makes these loops, and that also has a three-dimensional form, and that can also control expression. But the most important thing is that the binding of that protein is blocked by DNA methylation, so keep that in mind. And then so these loops are further divided into active and inactive compartments and then chromosome territories. Okay, let's have some fun with math. Now, this is meiosis. So this is the process by which we create gametes or sperm and egg. And so the first thing that happens before this stage starts is that your DNA will get duplicated. So you have a chromosome from your dad, one from your mom. So right before this, it doubles them. So then you have two from your dad and two from your mom, exactly alike. And then they come together, and that's what's called the tetrad. And there's a wonderful thing that happens called recombination, where it basically swaps genes. And that even gives it more diversity. But if you start doing the math just with the two parents, and without crossing over into account, you're already into the 70 trillion possibilities of chromosomes. And that's trillions more than the amount of people that have ever lived on Earth. And if you take into account all of these genes, these numbers can get astronomical. Although there are certain places that are more likely, they're called hotspots, to do that exchange, and some places that are less. So this accounts for the fact that all of us are completely unique, except, of course, monozygotic twins. Now, during that part where the chromosomes line up, again, we've seen this over and over again in nature. Sometimes it makes mistakes, and sometimes these can be helpful mistakes. Sometimes they can be deleterious. So the reason why this happens is because there's similarity in between the sequences. So it thinks that it's lining up in the right place, but it's really not. And what that can lead to sometimes is a duplication of a gene, and sometimes a loss of a gene. Like, for example, being able to see different colors was a duplication. We used to only see one color, and that got duplicated, and then we were able to see other colors. Now we're going to concentrate on this region that I was studying, on a specific chromosome 15. The region is called Q11 to Q13. And this is important because it's the Angelman locus, as Dr. Siegel talked about. And what happens here is you have these duplicons called HERC2 on these two ends, and they line up incorrectly, and you end up getting these type of deletions, with this one being the most common one. And this is a de novo truncation, which means that it's not being inherited from a parent. It happens during that process of meiosis. Now, this is important because this disease, Angelman syndrome, is a severe neurogenetic disorder that affects about 1 in 15,000 births. Now, this is due to that loss that I just explained, but only on the maternal allele. So this can happen on the paternal, and that's another disease called Prader-Willi syndrome. But if it happens on the maternal allele, you get this disease called Angelman syndrome, which is actually a lot worse than the other one. It consists of seizures. Sometimes they're resistant to medication. These children rarely learn how to walk. They rarely talk. But interestingly enough, they express this excessive happiness and a fascination for water. So let's take a look at this locus. And this locus is very important because, as you can see here, in non-neurons, in any other cell, UB3A is expressed from both the dad and the mom's gene. However, in neurons, it's exclusively expressed from the maternal allele. And that's due to something called imprinting or silencing, gene silence. And because those patients with Angelman syndrome have that loss of the maternal allele, in their brain, they still have that UB3A, but it's silenced, so they have no production of UB3A. And this is what we call in science kind of a low-hanging fruit, because you still have that gene there. But if we could just turn it on, we could tap into that UB3A as a source of UB3A, and maybe it could help these children, even if they could feed themselves, that would be a great step forward for these parents that take care of it. And so the silencing is known, the mechanism is known to be a long non-coding RNA, as I mentioned earlier, that starts at an upstream gene called SNRP, continues down a region that includes some genes called ITW and POR1, and transcribes antisense to UB3A. So if you remember, you can transcribe in different directions. This particular one transcribes, UB3A is usually transcribed in the minus direction, but this long non-coding RNA will transcribe in the positive direction. And it's that section right there that has been shown to be responsible for the silencing of UB3A in neurons. Now, if you look at the non-neurons, though, studies have shown that long non-coding RNA actually stops at a region around that IPWPR1. And serendipitously, there was a patient that was seen with a small deletion of that region right there, and they were actually expressing that antisense in non-neurons. So that indicates that perhaps there's the existence of some kind of boundary element that's lost during neuronal differentiation. Now, one of the geniuses of Dr. Siegel was that we noticed that in that boundary region right here, where binding sites for CTCF, And so we thought maybe these chromatin loops are playing a role in this silencing of this region. And so we came up with a hypothesis that CTCF creates a chromatin loop in non-neurons that prevents the extension of the long non-coding RNA past IPW and QR1. And so you have that loop there that doesn't let that RNA polymerase continue. And then CTCF is evicted by DNA hypermethylation. If you remember, when DNA is hypermethylated or methylated, then CTCF can't bind. And that's what's allowing that long non-coding RNA to continue past IPW. Okay, our lab specializes in something called artificial transcription factors. And this all originates from one of the greatest discoveries in science that was published in 2012. And this was one of my inspirations to continue into grad school, something called Cas9, which was originally actually a defense mechanism in bacteria for viruses. And the special thing about it that they found out, it's a protein that when you put it together with something called guide RNA, you can actually direct it to a region in the genomes, a very specific region. So think about it as basically like a shuttle or a taxi that you can tell it to go, I want you to go there exactly. Now, as scientists, we like to mess with things. So we've done a lot of different things with this Cas9. One of them that we do is basically create a dead Cas9 where we remove, I'm sorry, I forgot to mention that when Cas9 goes to that specific region, it cuts DNA. Now, that sounds like a good thing, but the evidence has shown that that's really bad, and all kinds of weird things happen. It fills in random things. Sometimes things get flipped over. So you don't really want to be cutting DNA as a therapeutic. So we created something called the dead Cas9, where we took that part of the protein out that does the cutting, but the function of working as a taxi is still present. And then our lab specializes in creating these fusion proteins where we actually add an effector domain to that Cas9, which can either turn genes on or turn genes off. And we can do that with a protein called DNMT3AL, which is a DNA methyltransferase. That means it puts the methylation. And another one called TET1, which is a demethylator. So we can actually change the epigenetic landscape there. So this was the aim of our study. This was our dream. I call it the CTCF eviction assay. And if we wanted to determine if CTCF finding at IPW and the QR1 is necessary for the termination of the antisense transcript by DCAS-directed DNMT3AL methylation of the binding site. So basically, we were going to try and methylate that region, keep that CTCF from binding, and see if we could turn a non-neuron into what a silent paternal neuron looks like, and see if we could see that antisense being produced. And the most important, the flip side of this, is the CTCF recruitment assay, where we want to determine if DCAS with TEC1, that demethylator, was sufficient to inhibit the transcription of that antisense past that IPW region in that silent neuron. So we were going to direct it to this region, hopefully hope that CTCF could bind there, form that loop, and we could actually unsilence that silent paternal allele, and as I mentioned before, tap into it as a source of UB3 for these patients. And Dr. Siegel was talking about this is one of the most important things in grad school, choosing your model. I knew I really did not want to work with mice or rats or anything alive. I really like doing cell culture, and we went back and forth. What are the things that are commonly used? SHS1/5Y cells, but these are derived from cancer. So they're going to have an aberrant epigenome. One of the most commonly used models are induced pluripotent stem cells from patients that have Angelman syndrome. But these are notoriously difficult to grow, and they can take up to 10 weeks to try and get them to turn into neurons. So the big advantage of these LUHMES cells is that they're these genetically engineered cells that were taken from the mesencephalon of an 8-week-old fetus, they're perfectly normal cells, except they were engineered. They inserted a gene called MYC, which is responsible for proliferation. So it keeps the cells dividing, and it keeps them in basically a pre-neuronal state. They're not neurons yet. And this is actually controlled by a switch that involves the antibiotic tetracycline. This is called a TET-off system. So when you add tetracycline, it actually turns off that MYC gene, and it allows the cells to go where they actually wanted to go and turn into neurons. And the biggest advantage of choosing the LUHMES cells was so it has probably a normal epigenome, and they take 7 days to differentiate into or fully differentiate into dopaminergic neurons. So that's a really huge advantage, because we can do a lot of studies on these and not have to wait those 10 weeks. The first thing we had to determine, though, is there really a difference? Can we use these LUHMES, the undifferentiated LUHMES, as non-neurons, and can we use the differentiated LUHMES as neurons? And so we measured the expression of the antisense, and you can see that the 7-day differentiated LUHMES were expressing the antisense, just like human adult brain tissue, and the undifferentiated LUHMES were not expressing it, just like another type of cell that's derived from the kidney. And then we wanted to see how that expression grew throughout the days in the differentiation median. And you can see that the expression of the antisense grows exponentially. Then we wanted to look at the RNA in this region. And a lot of times people do just regular RNA-seq. But if you remember, I mentioned that RNA does transcribe from 2 different ends. So basically, you're kind of hiding a lot of things when you do that. Luckily, our core, we were able to do strand-specific RNA-seq, and I was able to put this together. And you can actually see the direction in which these are going. So, for example, right here, you see the expression of UB3A. In this case, this is not allele-specific, so you're looking at the expression from the maternal allele. And just to clarify, LUHMES don't have both the maternal and the paternal still intact. So in that sense, it is a disadvantage in comparison to Angelman iPSCs. And you can also see that the transcription of RNA starts a little bit earlier than the undifferentiated cells here. Then we did a differential gene expression, and this is what we call a volcano plot, where this is telling the biggest differences that we see between the undifferentiated and the differentiated cells, and also our genes of interest in our region and how they compare. And this made total sense because these genes that are highly expressed up here have to do with neuronal function, and these others don't. And you can see that the genes of interest here are actually expressed more. So on the y-axis, you see the p-value. That's the statistical probability that what we're seeing is correct. And log-fold changes how much it's changing, whether it's in the negative direction or the positive direction. And here we wanted to basically see the different replicates, and this is what we call a heat map of these genes in our region. And you can see that they are in the neurons. They are expressed more highly than in the non-neurons. And have to give credit to AJ. He really helped me out with this. He's from the LaSalle lab. Now, what we do is we take all that information, stick it into a database, and it can actually tell by all of these genes, kind of group them together into functions. And these are called Go terms. And you can see here that when we did that for the genes that were upregulated on LUHMES, we find a whole bunch of neuronal functions. So that makes total sense. They are neurons. And the ones that are undifferentiated or downregulated, you see a lot of functions that have to do with like cells dividing and stuff like that. So this still kind of telling you that these cells are good for this type of experiment. And this was one of the tougher assays that we did, something called a high chip. And that involves basically kind of taking a picture, freezing the cells in time. Then we cut them up into pieces. We pull out the CTCF with an antibody. Then we take the CTCF out and just leave the little pieces of DNA there. And what has occurred is because you froze them in time, that whatever three-dimensional configuration that they were at that point, you're going to have the DNA from one piece close to the DNA from the other piece. So once you take, we've taken the CTCF out, what we do is ligate it, and then we end up basically with this hybrid of these two pieces that were perhaps, if you look at it linearly, far away from each other, but if you look at it in 3D, they were actually pretty close together. So we send that out for sequencing, and you can get these maps of loops that are happening. Now, our biggest shock when we got this back is that our hypothesis was completely wrong. In fact, instead of having loops that were present in non-neurons, we found two novel loops that were present in neurons. And you can see there's actually one that comes from this faraway gene called MAGEL2. And MAGEL2 has a function that's called an enhancer. This is another way of controlling the expression of DNA. And it acts as an enhancer to a nearby gene. So it's possible that it's also acting as an enhancer here in the neurons to get that long non-coding RNA to go express a little bit better. And then we also have that second loop exactly from that region, that boundary region, IPWQR1, and it goes to UB3A. So it's very suspicious that this might have something to do with that silencing of UB3A. Now, usually what we have to do in science is validate. And what we do is take another sample, do another assay, see if we get the same thing. And I really have to thank Nick from our lab. Spent many nights, very late nights, working on this thing. We had a lot of fun there, actually, but I do have to thank him. It wouldn't have been possible without him. So in this type of study, what you have is you start with a viewpoint. So this is, we were looking at SNRPN, and this has it's a statistical model, and it'll tell you if something is happening that's more than what you would expect. So it's normal that you're going to find a lot of the things that are close by that are going to be elevated. That's why you see these things here, but it's kind of counting them out statistically because it's saying, oh yeah, you're just close by, linear feet. But it did detect that MAGEL2 interaction as well in the neurons, and it did not detect it in the non-neurons. Now, let me see this. And then for that PWAR1 and the UB3A, we also saw the same thing, where there was that interaction, and we did not see that one in undifferentiated wounds. And then so our final assay was, we used a sort of new technology called Oxford Nanopore long-read sequencing. So usually when we send out for sequencing, we basically break up DNA into small chunks, and then we put it all back together basically like a puzzle. This Oxford Nanopore is unique in that it actually grabs an entire molecule of DNA, pulls it through, and it can read the electrical signal, and it can not only figure out what code of the letter that is in that DNA, but it can also tell by how high the electrical current is if that let's say it's a C, if that's methylated or not methylated. And that's what you see here. And I have to thank Ruda here from the core. She really helped me out a lot with that. And Danny from my cohort, who I'm sure all of you know, she's like bioinformatic genius. And I absolutely could not have done this PhD without her. Okay, so what did we get out from this? We were able to extract the profile of methylation. And I also found this kind of obscure pipeline where we were even able to separate the methylation between the paternal and the maternal allele. This had never been done before for this region. So this was kind of a really cool thing because we were going to be able to see what the methylation state was in that silent maternal neuron and what it was in the maternal neuron. And when we run statistics on this, because we have different samples, we can we call these things called differentially methylated regions. And what you see here, if it's elevated, if it's higher than 0, what that means is that it's more methylated on the paternal chromosome than the maternal chromosome, and this is in neurons here. And if it's below the bluer colors, since it's negative number, then that means that it's less methylated on the paternal neuron in comparison to the maternal neuron. And this is a comparison that we can do in both neurons and undifferentiated genes. And the coolest thing that we were able to do is we were able to integrate all of these things. If somebody now can look at this and look at the methylation profile, they can see how that's affecting RNA transcription. They can see where the loops are. So this can help people make better, come up with better hypotheses as to how to unsilence that paternal allele. And to get a closer look, some of the novel things that we're going to publish that we found, you can see here. So notice how in the undifferentiated and differentiated state, let's go back a little bit. If you notice, there's actually a loop, a little short loop that does occur in undifferentiated cells here. Now, if we get close to that MAGEL2, you'll see that there's hypomethylation on both the differentiated and undifferentiated cells. And you also have loops that land, so that makes total, complete sense. That's what you would expect. Now, this is what is a novel finding here. We actually found that there was a hypomethylated region in that SNRPN, and so hypomethylation allows that loop to form. So even though our high chip was not allele specific, and now one of the reviewers is asking for it to be, and that's something I'm working on right now, but we can also derive that because this is hypomethylated and there is the presence of that loop there, and this is hypomethylated on the paternal allele, that loop is probably only occurring on the maternal allele. Now, another really cool novel thing that we found is that close to that QR1 IPW, you can see here how close it is, we found a hypermethylated region. And so that's an undifferentiated cell. The other one was in neurons. This is an undifferentiated cells. So we think this could be playing a part in that boundary function. And it would make sense to also derive and say, oh, this is why we don't see it in undifferentiated cells, why we don't see that loop there. So it's possible that this loop that we're seeing here is also on the paternal allele only, even though we don't have that separated information. And you can see that actually there's not a lot of differences here on the UB3A, and except for that higher expression of RNA. Sorry. Now, you can see if you really map out here that there's actually, it's not a lot of DMRs that are called throughout the chromosome 15. That region right here where UB3A is, our region is really kind of a hotspot for those DMRs. So that's really interesting. So integrating all of that stuff allows us to kind of create a model that shows what we think might be going on. We think that these loops that are formed here are acting as an enhancer to that long non-coding RNA that starts at SNRPN. And so one of the theories about how this long non-coding RNA is silencing the paternal allele is called the collision theory. So imagine basically those two polymerases trying to transcribe in opposite directions, like two trains going towards each other. And normally they collide after UB3A has been transcribed. Then you get that production of UB3A. But our hypothesis is that with that extra loop, and some way this is involved as well, it's causing kind of like giving more speed to the train on this side and is not allowing that UB3A to bind, or knocking it off and not allowing it to be expressed. So in conclusion, we found those two new loops from MAGEL2 to SNRPN, QR1 to UB3A. We found that hypomethylated region on the paternal DMR at SNRPN. We found, and something that's exclusive to undifferentiated LUHMES that hypermethylated DMR near PUR1. And both of these are actually targets for those fusion proteins that we talked about, those artificial transcription factors, which would ideally be because we want to target the nervous system. So in order to recreate what we saw in the undifferentiated state, we could use that to methylate the neurons and try to undo that. Or in the neurons in that other region in SNRPN, also try to hypermethylate it and keep that one other loop from forming. So basically this shows that these LUHMES can be a pretty big bile. They're human-derived. A lot of research is also done with mice, and the region is quite different in mice. There are some disadvantages to it, so that's one of the advantages. And the big advantage, of course, being that rapid neuronal differentiation. And now we've given something where, like, look, this is all the information from these cells. Use them, take that, and run. Ideally, of course, we wanted to keep running with it. And so one of the first things that I needed to do was to create a model that was more like Angelman syndrome, where we would create what we call a knockout, or take out that maternal UB3A either by cutting it out or just stopping it. And for that, we use that Cas9, regular Cas9 that we were talking about that causes those double-strand breaks. But you can use a trick where if you add donor DNA, you can actually guide what the repair is. Sometimes it's not very efficient, but it can happen in some cells, and then you choose them. And in my case, what I had designed it is to put in, out of that code, a stop codon right at the beginning of UB3A, so that it basically would start to be expressed and then get stopped. So the problem is then, how do you tell which cells you cut? We can't mom or the dads, cut them both. You don't cut any. There's a whole bunch of cells in your culture. So I came up with a way that you could tell just by measuring the expression of UB3A in the undifferentiated state and then in the neuronal state. And you could see, because this is, of course, if there's no cut, you're going to have expression of UB3A both, because you're still getting that maternal one. If you have both alleles being cut, you would not see anything. If you have just the paternal, remember, the maternal does get expressed in neurons, so you would also see it expressed in both states. And this is the one we want, that if you actually got the knockout on the maternal, then you would see the expression of UB3A before differentiating the cells. But when you turn them into neurons, you would not see the expression. And the way we do this is basically you do cell sorting. We put them in this 96-well plate, and we try and do 1 to 10 cells in each little plate, and then we let them grow out, and then we test those cells to see these things. Now, one of the hardest things about these LUHMES is that it's notorious that they are difficult to get DNA into them. And I spent a lot of time working on this, a lot trying to optimize this, and finally found a way using electroporation that seemed to be efficient. And you can see here, this is me using a green fluorescent protein. And these are neurons. This is what it looks like under a fluorescent light. So this means this was a successful transfection, that I got the DNA inside the LUHMES. And I had all these plans, I had these elaborate plasmids. These are round pieces of DNA that we were going to use to test whether we could methylate or unmethylate and undo those loops. And I wanted it, I kind of got it ambitious and wanted it to also have a switch so that I could let the cells start growing and then turn the switch on, which would express that DCAS with the DNMT3AL. And then I could say that I'm not just blocking it, but that I could stop, or like it says that you could use it more as a therapeutic because you're actually intervening into neurons that are already formed or starting to form, rather than coming to the conclusion that you just stopped it from happening in the first place. Now, the disadvantage was that these LUHMES cells already have that tetracycline switch, which is the most common kind of switch. So I found this kind of obscure other kind of switch called a LUHMES switch. However, we needed to be in this off state so that it wouldn't be expressing, it was only be expressed when I add LUHMES. And it was very difficult to find the sequence to have it in that state. And trust me, I looked everywhere. The only place that really had it in that configuration was a somewhat sketchy company that we bought this plasmid from. So basically, you buy the plasmid that's supposed to be from like around it here from here, and you know the ends here, and then we do something called the cloning, which I'm sure every graduate student here knows how horrible that can be. Sometimes it works on the first time. Sometimes you can try for months and it doesn't work. And that what you're trying to do is, if you know the ends, then you can basically stick whatever you want, other pieces of DNA in between that circular DNA. So really, what I want to do is help other graduate students and leave behind the wisdom that I've learned. As you can see, that I did a lot of things and wasn't able to accomplish a lot of things. And a lot of nights that were spent that went nowhere. But I feel satisfied with what I was able to accomplish. So one of the things that is important as being a graduate student is to mentor others, to teach the next generation. And these are some of the important things that I've learned that I try to pass on. And I'm sure you've heard a lot of these things. Fake it till you make it. Work hard, play hard. Remember, it's a marathon, not a race. You got to have a good work-life balance. And as far as my experience, nothing ever goes right the first time. So expect that already. Just know that the first time you do an experiment, it's not going to be good. And just know that you're going to have to try it again. And that's really where PhD is. Figuring out how to troubleshoot. Trying to go back and see where that error occurred and how to fix it. And this is probably the most difficult one. Learn to let go. Accept when something isn't working and move on. And that really hurts because I forgot to mention, when we did this, I was trying to clone that DCAS DNMT3AL. I spent months doing this. I was there December 23rd, it was like 10:30 at night. I was supposed to catch a flight to go see my sister. And I was at the point where I was just about to tell her I can't go because I need to do this cloning. I need to get this cloning done. Fortunately, I was like, okay, it'll be there when you come back. But I was throwing everything but the kitchen sink at it. And it wasn't working. I was screening, and this process of screening hundreds and hundreds of colonies, running through gels, and why is this not working? Finally, a wise person told me, how about you send out for sequencing, full sequencing the plasmid that they sent you? And surprise, surprise, they sent me a junk. It was not what was intended. So obviously I didn't have the right ends. I didn't have the right design, and that's why it wasn't sticking. I would call the company, hey, you guys suck. Send me the right thing. They said, it's going to take 2 weeks. Okay. 2 weeks later, we still haven't been able to do it. And they strung me along. And then finally, months afterwards, they're like, oh, by the way, we can't do it. And that was probably the lowest point in my PhD. Because I had put in so much time and heart on this. And for it to be something that's out of your control, that somebody else screwed up and you've been working so hard because of that was heartbreaking. I'm sure Jeremy remembers that night that I called her in tears. And I'm sure every graduate student has or will have one night where he's with you calling someone that you care about. And so that was incredibly frustrating. And that's why it's important to keep these things in mind. So learn to let go, accept when it isn't working, just move on. Remember to celebrate those small victories. And like I said, lean on either your family at home or your found family here. And make it a story of determination, perseverance despite adversity. And most importantly, be nice to yourself. Like, we're all high achievers, are very hypercritical. And you have that voice in your head sometimes that's telling you, you know, you're not good enough or this is perfect. Just like that little voice should be your best friend. It should be telling you, encouraging you and telling you it's fine, everything is going to work out fine. Now I'm going to talk about my story. We'll leave the science behind. And as Dr. Siebel mentioned, so I'm a very non-traditional student, about as non-traditional as you can get. I was originally born in Nicaragua. My first memory is barrel bombs dropping on my neighborhood and us running through the streets with tanks and sniper bullets whizzing by us. Then we lived through a communist regime that was oppressive, and because my dad spoke out against them, they started to terrorize us. So we had to flee, and my dad was brave enough to basically sell everything we had and come and work in the States. And we were refugees, and these are only a few of the places that we lived because of his job, and we had to make a lot of sacrifices, and we moved around a lot. And that was also difficult at times because you constantly had to leave your friends behind. Once I graduated high school, and I graduated from Anaheim High School. If you guys know anything about Anaheim, it's all gangsters. These were my role models. If you look at pictures of me in high school, that's what I was trying to be. And I wish that somebody would have come up to me then and told me, hey, there's this career path, science, like you're good at science, you can take this career path. But, you know, my parents were immigrants. They barely knew that how to prepare us for college. Like, we thought high school was it. So once that came around, I thought I was going to magically go to Harvard or something. So did kind of like after I finished high school, we didn't really know what was going to happen. Through a incredible glitch, the Nicaraguan government messed up in their elections, and it was for a very short time a democracy. And so my dad decided to move back. And he said to me, you can come and study medicine over here. And I was like, I like biology. Yeah, doctor, I can do that. So went into medical school there, learned a lot, completely changed my life. And especially because when you finish medical school in Nicaragua, you go through this lottery system in order to be able to practice. And they send you to a random rural area to do something called the social service. And during that time also, unfortunately, we lost our mom. And I was very much depending on coming to the United States afterwards, after finishing that. And so I came and I was trying to challenge the board's exam, learn on my own while working. I did all kinds of jobs. These are just a few of them: medical translator, anatomy instructor. I was a server at the melting pot, making fondue at tables for a long time, probably the best money I ever made. A medical assistant. And when I was working as a medical assistant in a cancer center, I remember losing a patient that I had grown quite close to and just feeling that gut punch and remembering like, oh yeah, I don't like that. But I was still at that point where I thought, hey, maybe I still kind of wanted to challenge the board's exam. But my weakness is that in Nicaraguan medical school, it's very practical. It's very hands-on. Second year, you're already delivering babies, but very little basic science. I had only taken one biochemistry class, and that's what the board's exam is, basically, a lot of the basic science. So I came to a realization, and from what some of the doctors that I work with, hey, you have a lot more potential than this. And I realized I'm getting old. If I don't start changing things right now, I'm not going to get anywhere. I need to pivot. I need to make a change. And so I decided to go back to school, started at a community college, Orange Coast College. And it was at that point that I learned about this magical thing called CRISPR-Cas9 that Dr. Jennifer Doudna discovered. And I was so fascinated by it. And I was like, that's the future of medicine. That's what I want to do. I want to work with that. And there was a program called Bridges to Baccalaureate. And I was fortunate enough to then get into UC Irvine, which was totally my dream. When I used to be in medical school, I used to come to UC Irvine to buy my books. And I used to see these apartments that were right next to it. And I would dream of one day living in those apartments. And when I went to UC Irvine, I got into those apartments. So I fulfilled that part of my dream. The best part that happened to me at UC Irvine was a minority science program ran by two wonderful people, Dr. Marlita de la Cruz and Dr. Luis Mota Bravo, and they really inspired me. This program was dedicated to getting minority students to do PhDs, and they really are the ones that propelled me to come to UC Davis, and we'll see what is waiting for me, what the universe has for me from now on. Hopefully in biotech, but we'll see. And just a little bit, as you guys know, I said I'm from Nicaragua, and so Managua is the capital, and this is where I was sat to do my social service, which was actually originally another place around here until somebody asked me to go into the jungle and be their doctor, and they basically wanted to kidnap me, and my dad had to make some political things and help me move to a safer place. This is kind of what the town looks like. It's pretty cool because it's next to the water, and I can now boast that I delivered over 200 babies. And I just want to say thank you, gracias, Papa. This man right here is my dad, and he has his own incredible grad school story. While he was doing his master's in hydraulic engineering in the Netherlands, my mom was suffering from cancer. He was taking care of her. He was taking care of my 2-year-old sister. At one point, he sends her back to Nicaragua while he was getting ready for his final exams. And a humongous earthquake hits Nicaragua, levels the entire city, and he can't even get home. He basically hitchhikes like train, planes, and automobiles his way all the way back, not knowing if his daughter is alive. And then he comes back, and they give him a final chance to pass in his test and oral exam, and he passes it with flying colors. I want to thank my mom. I missed you so much, and I feel like every single thing that's good in me is because of this person right here. I miss her. And to my sister, my rock, has always been there for me, and this picture here tells you a lot about our relationship. She's always really loved me very much and been my protector. So thank you, sis. And to my mentors, some people are lucky to have one good PI. I feel blessed to have had two incredible, wonderful PIs that supported me so much. I really want to thank you, Dr. LaSalle. You're so compassionate during those very difficult times at the beginning. And I've always felt like you understood my strengths, my weaknesses, and you knew how to extract the best out of me. And Dr. Siegel, thank you for believing in me even when I didn't. And your compassion was truly the key to my success. Of course, I want to thank everybody from both of my labs, especially Austin Sharifi, who is another one of the smartest people I've ever met, definitely the most patient person I've ever met, and who I could not have done this PhD without. And Nick, as I mentioned earlier, to my committee, Kyle Fink, Dr. Kyle Fink, who was on my QE committee and provided a lot of great ideas. I also want to thank Luis Contreras Llano back there. He was my first mentor when I came here, my first rotation, and he taught me everything of the basic things that you have to do. And he was the first person that made me believe in myself. And to tell you something about Luis, I remember I was so grateful to him, and I told him, God, you've helped me so much. Like, how can I pay you back? And what he said to me was, pay it forward. Do it to somebody else. And Luis, I tried to do that. Thank you for telling me that. And I tried to leave your legacy behind. I also want to especially thank, I was very fortunate, and this is funny because it's kind of like this bookend thing. My very first roommates, Brittany Wanzer and Katie Dahlhausen, which you guys might know, she graduated from here. She did that famous koala study. They were my first two roommates, and we were just amazing together. We had so much fun, and they were just some of my favorite people in the world. And now that I moved to a different place, I have these two other wonderful roommates, Nico and Hina, and we have so much fun together, and we're such a good dynamic and family. And I just feel so fortunate, especially because at the end there, that final run, I just expected to be a whole bunch of misery and darkness, and they brought light to my life. So thank you, Mikir. And just remember, like, you're going to make some unbreakable bonds. These are some of the friends that I made along the way, not just here at this university. These are my best friends from medical school. I want to also thank the two people that were with me in my social service through some very tough times. I feel like I've seen more things in that year that any human should see. To my friends from the melting pot, which we had so much fun in it. Luis, again, Ile and Ram, some of the coolest people that I know. I mentioned my roommates. This is Katie here, and then that's Brittany. And this is just recently Nico. Hina and I went to do some wine tasting. So that's Nico and Hina. And of course, to the Calipages, this is what our cohort, 2017 cohort, dubbed ourselves. And if you guys that have studied in IgG, you know about the Calipage sheep, and you know the inside joke on that. But I was fortunate that we were a really close group, and that doesn't happen all the time. We really bonded, and I was really fortunate. And really, one of the main reasons why we bonded is and she's just an incredibly wonderful person and I feel like she was the glue that brought us all together. So thank you to all of the OPTs, and especially to Julie. And of course, my family, starting from our matriarch, my grandma, Yelba, my sister and her husband, Ryan, this agreement about that. Thank you very much. And their daughter, Taylor, my aunt, Nora, which I'm very close with, and her children, and even my pets. They put my pets here. This guy right here, he died in my arms, and he was the Scooter. He was the inspiration for me to go to medical school. I never wanted to feel that helpless again. So they're so much part of our families. And when I was in Nicaragua, I lived with my aunt Martha and her husband Rafael and my nephew George and our little dog here, Mocha. And it's just a really special family that I have, and I miss them so much. And I thank you for the support that you gave me just recently. I couldn't have written that paper without your help and without all of your love. And I just want to leave it with something. I'm a total nerd, and I'm a big dork for Dune. Read all the books many times, and this is what's called a litany of fear. And this is something that has helped me along, especially before presentations. I recite this to myself, and it really helps me. I must not fear. Fear is the mind killer. Fear is the little death that brings total obliteration. I will face my fear. I will let it pass through me. And when it is past, I will turn the inner eye and see its path. And where it has gone, there will be nothing. Only I will remain. Thank you, and I'll take any questions.
Good day.